चलो गाइस डीएनए फिंगरप्रिंटिंग का एनसीईआरटी रीडिंग करते हैं।
The technique of DNA fingerprinting was initially developed by Alec Jeffreys. He used a satellite DNA as probe that shows very high degree of polymorphism. It was called as variable number of tandem repeats, VNTR. The technique, as used earlier, involved southern blot hybridization using radiolabeled VNTR as a probe. It included I. Isolation of DNA, 2. Digestion of DNA by restriction endonucleases, 3. Separation of DNA fragments by electrophoresis, 4. Transferring, blotting, of separated DNA fragments to synthetic membranes, such as nitrocellulose or nylon, V. Hybridization using labeled VNTR probe, and Y. Detection of hybridized DNA fragments by autoradiography. A schematic representation of DNA fingerprinting is shown in Figure 5.16. The VNTR belongs to a class of satellite DNA referred to as mini-satellite. A small DNA sequence is arranged randomly in many copy numbers. The copy number varies from chromosome to chromosome in an individual. The numbers of repeats show very high degree of polymorphism. As a result, the size of VNTR varies in size from 0.1 to 20 kilobits. Consequently, after hybridization with VNTR probe, the autoradiogram gives many bands of differing sizes. These bands give a characteristic pattern for an individual DNA, Figure 5.16. It differs from individual to individual in a population except in the case of monozygotic, identical, twins. The sensitivity of the technique has been increased by use of polymerase chain reaction, PCR. You will study about it in Chapter 9. Consequently, DNA from a single cell is enough to perform DNA fingerprinting analysis. In addition to application in forensic science, it has much wider application, such as in determining population and genetic diversities. Currently, many different probes are used to generate DNA fingerprints.
ऐसे ही वीडियो और शॉर्ट्स देखने के लिए सब्सक्राइब करो बायो टिप्स नीट को।